7 minutes. Okay, that's not bad. All right, welcome everyone. I'm Shyam Prabhakar from GIS, and
Yeah, I'm Oliver Stegle from Heidelberg EMBL and DKFZ.
all right, we got a mission and scope here. We're going to try to reverse engineer this session. So, yeah, I guess you can still hear me. We want to come away from this with some practical projects that hopefully you will all contribute to and you all find interesting and valuable for our mission and scope. Right, so yeah, because we have very little time, there's so many exciting topics to discuss. We're going to try to cut all of that short and narrow the funnel down to, okay, what are the really important pressing things that we need to do now in the short term, and then much more long term, maybe a more general discussion of what's the ultimate goal, right? So very briefly, we want to discuss algos, pipelines, data analytics SOPs that we need to support the overall goals that Holger laid out and the other speakers laid out, right? What are those overall goals for the spatial program in HCA? We want to support healthy atlasing, and we want to support disease applications of the healthy atlases. And we want to do this globally, all over the world, so that we provide benefit to diverse communities all over the world, and along the way understand some science also of how human beings are different from each other, right? So three pillars basically, or three goals of this spatial program: healthy atlasing, disease applications, and how diversity plays into both of those things. Uh, any quick, and then I mentioned metadata specs also, and that has come up a lot today. We're still just about sorting out what metadata we need for single cell studies, and we've made a lot of progress in the HCA. But then for spatial data, again, there's a whole different question of what metadata are relevant, particularly technical metadata. But also, if we're talking about disease studies, what disease metadata we need to make these things possible. And this is really ambitious. What we're talking about here is ultimately some disease applications to specific diseases to derive biomarkers and drug targets and so on, right? So, very broad topic. Any questions quickly? Ollie, you want to comment on the scope, or any questions on the scope?
No, just one suggestion, which I think particularly could come up in the benchmarking aspect. I think we could also express characteristics or wishes how benchmarks could be tagged on to the data generation efforts. So I think if we have such suggestions, that could be quite useful, because benchmarking is obviously a key bottleneck in need. And I think if we come up with, a dream benchmark data set, we could express that, and then we can see what's feasible later on. I think that could be a very practical outcome of this session.
Any questions about the mission scope?
I'm not aware of any. I'm not aware of any method related to.
I don't know if people online can hear you.
Hello.
Not yet, not yet. You just, yeah. Okay, now, yeah.
Hello. So I'm not aware of any method related to spot detection for the spatial. So if you have any publicly available algorithm or tools, so can you comment on that?
Sure. I don't have spot detection in the list. We've played with spot detection algorithms in the past ourselves. Right now, we're just using what the manufacturer gives us for spot detection. Ollie, you want to comment on that?
Yeah, it depends between the assay. Yeah, I think that's where really different assays are quite different. So we have some work on that on the space, but I would maybe have a meta comment to that. Maybe it would be useful to think about what could be recommended solutions for different problems, and then spot detection would be one of them. I mean, I think we should sort of figure out what's the consensus, what are the solutions are. I think spot detection is an important problem still not solved, particularly for then the sort of more homebrew methods like Merfish and others. It's really a not trivial task. I would agree, it's a worthy problem.
Thanks.
Yeah, a very interesting problem. And I've listed, we'll add it to the list of challenges over here on the slide. But then hopefully maybe by 5 p.m. or something, 5:15, we want to funnel it down to, okay, which is is spot detection the most interesting problem? It's in one of the 3 or 4 most important problems for us to address algorithmically. How much better can spot detection get? Surely it can get better, but for this team, is it the one or 2 things that we want to focus on initially? Yeah. So that's a question. I'm not suggesting an answer to that question. Yeah. All right, I guess everyone else is okay with the mission and scope. Okay, let's get started. Ollie, you want to overview some of the challenges that and your priority list?
Well, I think we should have a consensus on a priority list. Maybe we could even ask this, what we should first focus on. And it would be great to hear from everybody in the room what you think are the most pressing needs. And we could maybe start low level and then work on to a higher level analysis and think about where the most important challenges are to overcome for our task. And I think the sort of suggestion for structuring that could be that we have on one hand a element on representing data in unified solutions. Then we could think about raw processing, spot detection, stitching, segmentation, and then we could do downstream analysis. And I think it would be really useful actually get a bit of a flavor or a sense of where people think this group should put effort on versus where we build on existing solutions or don't put efforts on, if that would be useful. Do we show of hands to get a sense?
For specific topics,
Yeah.
Specific challenges.
Yeah.
Yeah, let's maybe break it down into. So we have these 4 categories of algorithmic or data analytics challenges. So perhaps start with the first one, which is general spatial omics data analytics challenges, right?
But if within that, right? I mean, there's a question a little bit about what people want to talk about or what you think is useful. So the sort of low-level cell segmentation, quality control. Who thinks this is one of the most pressing needs? Okay. Yes, I also think that. It's still a small group.
I think it'd be nice to have some community hackathons like around some of these topics, like comparing benchmarks and stuff.
Yeah, that's precisely the kind of outcome and project we want coming out of this breakout session. I would certainly support a hackathon for cell segmentation.
Hackathon for cell segmentation. There's some questions over there, maybe a microphone. I don't know whether we do this or do we give up. It's just killing the whole dynamics, if I may say so.
So there's been, I think, there have been, at least in Germany, there's been 2, I think also with international participation, hackathons for cell segmentation and other pre-processing challenges in the past 2 years run by Elixir. And that's a European network, I guess, right? But I think tracking, like kind of piggybacking on something like this on a wider scale might be worth it. And this is organized by people in Berlin, basically.
Right. There were some comments here at the front.
Esther, do we have only one microphone? Yes. Okay. Then, if it's, yeah. Any other comments? Someone at the back, yeah.
Benchmarking on spatial data integration.
Great, thank you. We have benchmarking as a very broad category. Yeah, spatial data integration is certainly one of those we want to benchmark. We also want to benchmark cell segmentation. I would say, actually, we want to benchmark everything in that list. So the question is, so develop algorithms for everything in that list, and of course, if you're developing an algorithm, you have to benchmark it. Yeah, so, and then Ollie brought up the topic
but I think there's another comment there, but maybe...
Lucia? If you shout it out, I can repeat.
Sure. Oh. Hi, everyone. Hello? Hi, everyone. So there's a couple people online. I'm going to be the voice of the virtual audience. First comment from Dana Paya. She thinks it's very important to work on the low level and the high level concurrently. There is garbage in, garbage out, and I think segmentation is still in the garbage level. But then nothing is ever perfect, so we need to know the impacts of imperfection downstream to see where low-level efforts are needed. Also, downstream methods will need to take into account imperfect low-level stuff.
Absolutely. We often rewind to that's why we started with cell segmentation, data, QC, feature selection, so on, where the very early stage things. And everything that builds on that can be weak if you don't get that right the first time.
Yeah, on that note, I just started on spatial stuff, so I've been asking around about best practices of QCing data. I have seen very little consensus about this, so I think it would be very good to address this.
Thank you. Data QC SOPs for spatial, important, not important? Okay, raise your hand if it's important. Oh, wow, great. Yeah, and you're right, there's very little on that. So specifically, what we've seen is the obvious things for data QC are throw away bad cells, throw away bad spots, and then you're still left with things you want to throw away. What's left is some doublets, segmentation doublets. So some are bad, meaning they're low quality, and some are bad because they're actually artificial doublets created by cell segmentation. Those would be two obvious things in data QC. And then there's the higher level QC, which is throw away an entire bad data set, throw away a bad field of view, and so on. Malta?
I think the challenge in spatial QC specifically is the context matters a lot. So if you can't just remove one cell, and then basically algorithms that look for a spatial proximity need to be aware of this. So it's not as simple as in dissociated data where we kick out a few cells and they don't really have an effect on the rest unless it's a super rare cell type. But the downstream analysis that will be done will often rely on the spatial context. So how do we develop QC approaches that take into account the effects of that missing context if we remove these cells?
Excellent point. Your downstream analysis has to be robust. Already, even starting with Visium, there would be spots that you throw away, right? So definitely all the algorithms have to be comfortable with patchy or somewhat patchy data. If they're not, then it'll be very hard to work with. Let's go down the list quickly.
One thing I think that would be really useful is to maybe this prioritization a little bit. What I gathered from just the show of hands and the responses was that the low-level aspects from processing, QC, normalization was considered to be quite important. Would there be consensus that this is sort of one of the priorities we should do? Yeah. Okay. So maybe we have that. Then there's one element that we have not mentioned, which is the experimental design. So how do people think about markup or panel design? Is panel design now no longer an issue once you have 5,000 probes, which you now have? Or what would be there the take? Because that's a whole field of its own right. And I think there is a question to what extent and how deep we want to address that.
Sarah?
Yeah, I can also just, so I would just say it depends on the experiment, and I would say it depends on the question that you're asking. So if your question is about, let's say, a specific disease cell state that has this protein expression, then obviously having that in the marker panel is crucial. But if your question is more about, what's my tissue architecture, is it conserved kind of in this condition versus this condition, then it doesn't really matter. And furthermore, also where we're really advancing, I think it was in one of the talks, is the imputation based on marker panels kind of out to the whole transcriptome if you have a paired reference data set. So I think in that sense, it really depends on the experimental design, the question that you're asking, whether the marker panel, how important is it? Having said that, the ideal kind of dream is obviously if you don't need that, and you have full transcriptomic breadth at single or even subset, single cell or subcellular resolution. So, yeah.
Yeah, very true. To use a GWAS analogy, the more markers you have, the more in your panel, the more other markers will be in LD with a marker in your panel. Yeah. Gary?
Even if we don't need to define markers in that way for like cell types, I think we need to learn more about markers from a spatial perspective, because markers can have spatial characteristics, spatial patterning, and that is not observable from single cell RNA-seq. And so some of the cell type markers from single cell RNA-seq might actually be, like, I'm sure the brain people would have lots of examples where all the different subtypes of neurons actually have spatial components, and some of the markers are spatially variable among the, in special ways. So we need to probably learn about that as a new type of information associated with markers.
Absolutely. So it's a new kind of correlation that you can't see in single cell data so readily, meaning you could have the markers correlated in this part of the tissue, but globally not particularly correlated. And single cell, you cannot get that from single cell data.
Sort of, you can.
Sarah doesn't agree.
Gradients.
Lucia.
Hey, another comment from Dana. She thinks that 5,000 probes is not really a solution. It comes at a high quality for the genes you want, She dislikes the 5,000 probe set because for the given tissue, it includes many genes that are irrelevant. And we did benchmarking and it comes at a very high cost in terms of the quality of the 500 or 5,500 genes you care about. So for now, where the tech is, we still need to design panels. The high quality cost trade-off. That's what her first sentence was. Sorry. That's all.
Excellent points. Ali, you want to address it?
Yeah, I mean, I think we maybe have a few more comments if they're still on the panel design. I think it was just a probing question, but that's still on because nobody mentioned it. So I think we have it on the list. So that's maybe enough already. But if you have a few more urgent points, that'd be great. I think Sheila, you're next.
Yeah. Thanks. Just riffing off what Sarah was talking about, from making the panels, we end up imputing gene expression or any other sort of quantity. We end up with these sort of extracted features, or these are not necessarily the observed units from the data assay. And that's great for visualization and so on, but then, if we're performing modeling afterwards, we want to be a little bit aware that this is actually extracted features. So maybe something that we could think about is, how do we keep that provenance within the spatial program? Maybe that's something to think about.
As in how the markers relate to the features.
I use the V1 single cell atlas to impute the, V0 spatial. How do I know that those genes that I'm getting out are pretty good? Like, can I actually put some confidence on it? What drove that prediction? Because at the end of the day, it is a prediction. So maybe all the things that make statisticians a bit uneasy about using point estimates of a prediction, maybe we could try to bring that in.
I guess you're talking about imputing spatial information from single cell data?
Yeah. So, we're using a panel and then we want to get a whole transcriptome output, which, many approaches do this. And I can see a world where we want to include that within our HCA spatial output. So yeah, keeping that in some way.
Exactly. So that comes under this very broad rubric of correspondence to single cell data and integrating single cell and spatial data.
But I hear actually 2 points, if I may. One is provenance tracking of the data used to derive marker genes, which I agree is really important metadata. We should maybe put this in. This is under the metadata category. And the second point you mentioned is that the integration with corresponding single cell dissociated data should be also one priority, given that the HCA has generated so much of that already.
Oh yeah, for sure.
So there's one more thing which maybe I misunderstood, but the way I understood what you meant was that if you do an imputation, you keep track of where the model is imputing from and what's the confidence of the imputed. So imputation into the spatial from the single cell, not just imputation from the single cell. You know what I mean? The other way. I mean, both ways.
Yeah, so you can imagine keeping like a graph of who are your nearest neighbors, right? Like, could you have a graph representation of, for every spatial cell, who are its nearest single cell?
But that boils down to essentially having a uncertainty-aware imputation methods, right? I mean, that's a property of the imputation. I agree that should be a research area. I mean, maybe we could go yeah.
And then also what data have you used
Yeah.
to build that model?
Yeah, but I think that it's complicated, particularly if the marker genes are sort of dependent on the data you use to impute, then it becomes really circular, and I think that's an important problem we should keep hold of.
So I think, yeah, just to summarize this, 2 points that came up, which I think we should note down here. One is gene panels. Generally, so many existing technologies are panel-based. So as a community, we just need to, and if you want to generate data with those technologies, the first question you ask is, okay, which gene panel? And then you do it, you generate a big data set. 1 year later, you say, oh my goodness, we messed up. We didn't have these genes, right? So I think this defining gene panels is something we do need to have in our priority list as a community. And then once we've decided the gene panel, different people will have different gene panels. So reconciling and integrating across data sets from different gene panels, or data set with your XYZ gene panel with specific single cell atlases, I think they should both be in our priority list. Well, and I would say integrating with spatial single cell data should be in the priority list, even if we're not doing panel-based technologies, even if we're doing sequencing-based technologies. So yeah, I'm glad those 2 points came up here. Malte?
Yeah, I think that last point about integrating cross panels, I would put that as a higher priority than the marker panels themselves, because marker panels will inevitably have to be technology specific because of different requirements by technologies. And in the end, the companies have the power there because we don't know all the requirements for building a marker panel. We've been working on developing a method for this for 3 years. It's nearly finished, but there's just some things we cannot take into account, whether we can build this, target this gene in this technology because we don't know what the requirements are for that technology. And so while it's useful to have like general marker panels, if you adapt them to any individual case, they will always have to be modified, and then the panels you generate will again be non-optimal. And so I think we have to have some flexibility for what panels, what generate data that are there. So I put more focus on this integrating across panels, because realistically, we're going to get data sets from a lot of different panels. And that if we can manage integrating those, I think that that'll be very powerful to build a bigger reference.
No question, we have to be able to integrate across. Nobody's all going to use the same panel for various reasons I mentioned. But I'll go back to one thing. One of our aspirations is that each bio-network will define at least one set gene list that all the companies should include to the extent possible in their marker panels, because that will make our lives easier. Like you said, those panels won't overlap 100%. But if we as a community put out publicly reference lists of panels, it'll make everybody's life easier. It'll make the company's life easier because they'll have a starting point. And then when we buy those products and use them, it'll make our life easier because my data set and your data set will be at least reasonably similar.
We ask the companies to share the requirements for the panel design to be able to generate that, make it realistic.
Yeah, thanks. Yeah, I do wish we are hoping this will be a collaborative thing. Yeah, but of course each company will have different requirements. But at least we can say this is our master list of all the markers we would like to see. And then we can bioinformatically shrink it to some achievable number. But at least biologically, these are the cell types we would want to, and cell states we want to characterize. There's a full list of good markers for them. And now let's decide what is the best subset to put on your panel.
I have one more thing to add here. So maybe on the QC side, which I think is missing, is like I think more maybe not naive, but like more. I think at this point it might sound trivial, but which is not. Like on the computational side, how you define spatial domains, like what is a niche, and then that's also directly connected to the panel design too, because the number of features that you have, it can also affect how you define a niche or a spatial domain. Because I think one thing like that I see in spatial data analysis, which might not be like as prominent as it is in single cell, because with single cell, let's say like at beginning of the analysis, when you create the KNN graph, the K will not kind of decide whatever you do the rest of analysis, and it's more robust because we can go K equal to 50, you can still find your cell types of interest, whereas where spatial data K equal 4 to create a graph, and whatever downstream analysis can change everything downstream of that. So I think like how you define that relationship of like this neighborhood analysis and how you define the niche, plus if you integrate across multiple data set, there's a relationship between like using overlapping panels too, right? Because decay, how you define niche, directly connects to the number of shared features. So I think like just an elaborate discussion on this would be also super cool. Yeah.
I'm trying to distill your point. Do you have a specific suggestion?
I think if one can, I think it's going to be impossible to define what is a niche for spatial and then define a like specific case or range of case. But I think it would be good maybe to sit down within like different tissue groups and come up with a gold standard range for different tissues to see how they define a spatial domain.
But more, I think there's a more general point that you mentioned, which I'd like to ask a rescue from this, which I think is really interesting, that because the marker panel sets are smaller than the single cell RNA-seq, quite a number of the analysis steps we do canonically are not quite as stable. Yeah? Like building a KNN graph on 100 marker genes, just a completely different beast than 10,000 genes. If you integrate this across data, it becomes very complicated. And I think just revisiting the sensitivity of many key analysis steps in a single cell field to this small p, right, where we no longer have large p, that I think that's an interesting question, which I think is a really good idea. And I think that embraces many of your points, right?
Yeah.
Lucia?
If there's time, from Dana, if we want to make an experimental design recommendation, I think we should include a request for some amount of sequencing-based data, e.g. Visium OpenST, so that we can have some genome-wide spatial data as part of our inference and atlas building. While we can do a lot quite well with disassociated single cell and smaller panels, some measurements need to be genome-wide, and these can play into the inference to all data.
Absolutely. We don't want to give the impression that it's all about panels. Sequencing-based technologies are an important complement. And you brought up tissue segment. What I had written on as tissue segmentation, so I meant tissue domain segmentation. Thanks for raising that. Last couple of things on the general challenges side before we move to benchmarking, disease and diversity. Cell signaling, important, not important? Inferring cell signaling from spatial. Malta is nodding yes. A couple more people nodding. Okay, I'll take that as important. Are people raising their hands to say it's important or raising their hands to ask a question?
I want to ask question.
Okay.
So in the gene panel, whatever question it was arises, so it is possible to include the ligand receptor gene in the gene panel list so that both types of gene can be equally
You want cell
co-detected.
type defining genes in your marker panel and also biologically relevant genes, for example, ligand receptor signaling, for sure. Yeah. Okay, so Ollie had some thoughts about benchmarking. Maybe segue to that.
I mean, the one point that I think would be very useful for this community is to think about, at this early stages now, recommendations or wish lists for adding some benchmark components to the data generation aspect. This might be cell membrane stains, selected data sets for cell segmentation or other dimensions. So I think it would be now a really good point to think about these would be the dimensions we think would be important, maybe not in every data set, but some. Dana mentioned parallel sequencing-based assays, which is great as well, because then you have a comprehensive view, maybe not at the single cell resolution, not quite subcellular and so forth, but you can obviously definitely cross-check things. So I think if you could now come up with some of these ideas, so what could be in one or two contexts be a good benchmark utility or a data set or characteristics of data that would help our efforts, that could be a very helpful.
We've been thinking about this a little bit in the context of open problems in single cell analysis. And I think one of the hardest things in my mind to get ground truth for is this kind of downstream like niche detection question. And so it would be great if there was a benchmark data set which included, let's say, some spatial data sets with orthogonal clinical metadata so that you can assess whether the niches that you define are meaningful to predict this outcome because we in the end expect some kind of niche to be a functional unit that matters towards the phenotype of interest, right? And so if we have matching metadata that captures that, we could really enable a comparison of different ways to define units within a spatial data set that matter towards this prediction. This could be clinical metadata based on outcome of a disease, treatment response, all things that we think spatial co-localization or organization of cells should be predictive of on some level.
Can jump in. I mean, I think just having this metadata would be useful. And I think it depends on the disease group, what they use for. I think pathology is probably a modality, but I think having that request is a great idea to add that in.
Definitely pathology data, IHC data, some kind of independent data modality for as a benchmarking your domain or niche segmentation algorithms, very valuable. Dana?
We need to have a data set wish list, but our job is to think of the right benchmarking metrics. For example, too many of our metrics are collapsed to cell type, which ignores important variation within cell types. So as a community, defining the right metrics is as important as defining the right data sets that have some degree of ground truth to them.
Very much, yeah. And that's the thing about spatial. There are so many ways things you can infer from the data. Which specific metrics was she referring to? What kind of metrics? What was the objective? Okay, while we're waiting for that, so we don't have time to discuss in detail everything here. Maybe what I'll suggest is if you could read all these things. So definitely we talked about a little bit benchmarking. For disease, new questions come up, which is your tissue. So Grace showed a nice example of the tissue being the colon tissue being disorganized in the tumor relative to the normal. So that's a question you can't ask with single cell, but it's definitely an algorithm. So what is the tissue disorganization equivalent of differentially expressed genes? How do we even define that, right? Differentially organized tissues. So how do you quantify that or define it? But I think it's an interesting and important question when we get to disease, because it's not simply that the same cell types are expressing different markers, because that's a D gene question. And then there's the more downstream question, which is discovering biomarkers. These could be diagnostic markers or targetable biomarkers. And there, what we're talking about is associating what you see, something you see in the spatial data. It does not have to be a molecular abundance. Right? In single cell RNA-seq, we're all talking about molecular abundances, cell type abundances. Here, it could be a molecular spatial pattern rather than a molecular abundance, but still correlating that with your outcome, survival, drug response, your disease subtype, things like that, right? So definitely biomarker discovery brings up new questions for spatial disease analyses. And then somebody mentioned integrating with digital pathology. So that is a big diversity and benefit question for us. Aviv has mentioned it before as well, which is, can we take H&E as input, for example, and predict bulk RNA-seq or predict a spatial molecular profile? There are already algorithms to do that. They can be made better with better training data. And there is an immense social benefit to that, because in many parts of the world, molecular diagnostics for cancer, for example, are simply infeasible. The nearest lab is hundreds of miles away. But for molecular diagnostics, but for whatever reason, digital pathology is much, much more accessible, even in low resource countries. So if we can take that available digital pathology data, turn it into at least some crude molecular diagnostic using AI, immense benefit, and that's something HCA can contribute. Okay, so that's some topics in disease, and then of course signaling interactions, inferring. We want to see again how has signaling been dysregulated in disease. One question there.
So it's maybe like too naive, but in my opinion, I think it's also very important to consider across tissue types, not just focusing on like atlas for different organs, because one thing that I think would be very important also for the non-computational community is if they find different niches that are distributed in a similar way, that have probably similar cell types, are they behaving in the same way in that organ as they are over there in like some other distant organs or not? And maybe one of the things that one would consider is to try to also functionally identify them, identify these niches, not just, because I mean, like from the discussion so far, it appears to me at least that most of these efforts would be diagnostic at best, but nothing that would, at least in the near future, facilitate target discovery.
Yeah, I think, I guess you're saying that detecting these things across multiple tissues will help in target discovery? Or did I misunderstand the question?
I think we should like consolidate what we're doing. So like if we're, instead of just thinking of running it for only one, like how I say it, we're running each one of them separately, like one at a time. But maybe we should use like prior knowledge that we already have, and then try to just, you know, like compare across the different tissue types, because this I think would have a huge impact and also would help a lot with like people being able to run diagnostics, for example, in areas where that would be very difficult.
Got it. For example, lymphoid aggregates can be there in tumors from many different organs. Yeah, that's a good point. There's a question here.
Yeah, I have a question. Basically, it's about how you pick the region that you analyze. So how big, which on one hand probably depends on the question, on the organ, and is constrained by the technology. And in a way, this also may affect how you do some of this analysis of niches and so forth. And related is if you are trying to bring together different samples, even if it's the same organ, but you know, from how they were collected, maybe let's say you have more or less blood vessels, or blood vessels are elsewhere in your sample, and you need to do some type of alignment or some type of analysis to be able to compare. So all that questions of architecture and how you take that into account.
I guess you're suggesting we could come up with algorithms to choose the region of interest that we profile with spatial.
Or given that would be one, or given that you have samples that you got, let's say in one organ, in the gut or so, how do you know if they come from different patients? I mean, this of course also would be the case with only single cell, but how comparable are the different samples from different patients? But with the space, you have the additional information of, the architecture, and then, you know, if you want to, I don't know, compare cells and, they are closer to a blood vessel, how you take all that into account in integration. It's quite vague.
Sarah?
Yeah, so I'm not sure what Julio, like I'm not sure I got 100% the gist, but in the recent paper that we had in bioinformatics where we published the bin to cell and the gene to probe, one of the things that Nadav was playing with there is basically, you know, imputing the single cell transcriptome from the cellular morphology in the H&E, so it's kind of what you're saying. And then what you are saying about regions is then, of course, that allows you to extrapolate out. So let's say in a 2 centimeter by 1 centimeter H&E slide, but you've only profiled one small region with the expensive Visium HD, like 6.5 by 6.5 spatial transcriptomics. You can then extrapolate out the rest, you know, based on this kind of principle, because it's a repeating architecture, and then you can extrapolate the repeating whole cell transcriptome. Do you see what I mean? Like from the...
But I guess maybe to elaborate or to make it a bit more, this I agree, no? But if you make it more concrete, so let's say you have 2 samples from 2 patients, supposedly the same organ, but maybe in one case you have cover more or less part of different regions of the tissue, or you're closer or more far away from different places.
I mean, I think that very much ties into the need for a multi-scale representation of a tissue, that you have sort of organ-level characteristics you can take into account when you look at the macro structures that you actually then capture using a high-resolution assay. I think that's very interesting. I wonder what that implies for metadata that's also assayed. I mean, you almost wonder, do you have the metadata on this macro scale from the tissue you then have? And I think the sort of landmarks of where was the tissue extracted, that we can do this inference, that would be important, and maybe we could note that and bring this to the experimental groups as well.
Yeah, I mean, what's worth saying is that Gary and I are co-directors on the Canadian Institute for Advanced Research Multiscale Human, sort of program. And one of the, to link basically in a quantitative framework where single cell and spatial data is coming, in this multi-scale kind of philosophy, what we're doing is, for instance, linking these large-scale morphological measurements like hierarchical phase computational tomography, where you capture a whole kidney 10 centimeters diameter at 2-micron resolution, so you can sort of see the morphology or the texture of every cell. And then from the same kidney, kind of probe from the inside to the outside, where you have we've shown you have this immune kind of gradient with Meneklatworthy and Moos and Sambisrati and so on. So you have a gradient that goes from the inside to the outside on the kidney. You also have a salt gradient that kind of goes around the kidney. And so then, if you model that as a gradient, you then, sample your spatial data sets and you have the whole kidney, you can then model these together, if that makes sense.
And I think the multi-scale aspect is really critical. You can consider it across scales, like in cells, we all know how to recognize mitochondria and other organelles. It's the same problem. So, that problem is probably going to be replicated across scales. And, it might be nice to have computer vision people coming in to help with some of these things.
Can you define a specific multi-scale problem statement?
Just to consider multiple scales when we
I see. More about a strategy rather than a specific algorithm to be developed. Got it. Yeah. Very, very relevant. So I wanted to switch gears because we have 25 minutes left to the MVPs. But just before that, any important, really important challenge that we just have to address that we've left out in this part, in this section? Over there.
I can speak up. So are there just very basic spatial features that everybody should be computing in their spatial data? Like, for example, I can imagine a matrix where you have cell types versus cell types and their interactions. And it seems like if everybody agrees to produce that kind of a spatial feature, those things can also be used to define tissue regions because they share this, a region might share the same kind of interaction feature space and things like that. If we could come up with just some minimum basic features that everybody agrees to compute, that might be helpful.
You want to take it?
Sorry. Can I think it's a great suggestion. It also goes obviously in the differential analysis, right, to be able to do that.
Yeah, then you can do
But
differential analysis.
I think it's one of the areas how to do fingerprints from spatial data sets that one can use to characterize them, and then also drive differential analysis downstream. So I think it's a valid point, and particularly sort of the idea of having simple, among, in addition to sophisticated method, also simple characteristics that are easy to drive.
I guess
Thanks for that,
something
Marta.
that follows on from what maybe Julio was saying before a little bit is we seem to be missing common coordinate fragments in image registration. I know that was the topics in past meetings, but it seems like it's not really solved yet. Maybe also computational image registration, if we can kind of predict from what anatomical location, if we have some reference, a particular sample came from, to get higher quality than just left lobe, for example, in the lung.
That's a nice one to add. Imputing missing data, missing metadata on which part of the tissue this came from. Yeah, that's a great suggestion.
Just one more comment.
Yeah,
also, regarding the differential spatial organization, I think also some algorithms are just based on presence or absence of cells, for example, defining niche, defining neighborhoods, but also I think there needs to be attention to the topology of these cells, the actual spatial organization. I think there's many algorithms, mathematical algorithms that are used to define geometry in these cells. And I think that's something that can relate to immunohistochemistry and AI detection.
Yeah, thanks for that. I think the current algorithms, there will be some graph structure that the graph convolutional network or equivalent method tries to optimize. But those graph structures are not necessarily easy to interpret. And then, like you said, the non-neural network methods will do something fairly simple, like, okay, your domain is a region a coherent region that has this cell type composition, but then you're losing the topology of how the cells are organized in that region. So I would say, yeah, maybe topologically aware domain segmentation is a very nice algorithmic problem statement. Thanks for that. Okay, let's, one more question.
Sorry, I have it's a more general question, but are you considering taking gene biotype into account? Because so far you are using gene as a global term, which I think is fine, but there are different types of genes, the ones that are producing protein and the others are not. So the difference in the final product of gene expression is highly impacting the analysis that you are doing. So this is my question, whether you are planning to address the challenges and the opportunities by looking at different gene biotypes.
I would agree. It's particularly relevant, obviously, if we have multimodal profiling, we have protein imaging. So I think the multimodal dimension is interesting. I mean, one thing that reminds me of this, and I wonder whether that's something you're implicitly hinting at, is whether we want also to learn, sort of gene embeddings. Basically, before we've been really good at embedding cells, right? We've done that for many years. Understanding where cells are, and I think we now will also learn a lot about the relationship of genes, which also goes to the relation to fingerprints. I think some notion of this field being now enabled by spatial data would be very suitable. I'm not sure that addresses what you suggested, but there's maybe another comment.
I would like to add a question about these disease-associated markers. So, because I think the ultimate goal of this atlasing and these references allow us to compare disease cell with healthy. But this question, I think, is still largely explored because there's an inter-patient heterogeneity. When we want to compare disease with normal cell, which cell to compare is still a problem. We don't know whether the adjacent normal cell, or maybe the different patients, their disease cell have different developmental origins. So I think we need to, as a community, establish a framework or something to provide a guideline for the comparison and identify disease-associated markers.
That's a great point. It comes to our atlasing. If we just have a healthy atlas, there will be all kinds of cell states we see in disease that are unrecognizable, and the correspondence to, we won't know which healthy cell type it corresponds to necessarily, Gary. Gary over here.
I just want to mention maybe an interesting algorithmic thing. I don't know if anyone's working on this, but you could imagine like the analogy of trajectory inference over space that takes all the spatial images and infers the developmental spatial changes, because we can't measure time series with spatial, but you might be able to infer that.
That sounds cool.
Challenge.
That sounds cool. The spatial trajectory of the tissue architecture over a disease course or a developmental course. Wow, nice. Okay, quickly, we have only 15 minutes left. So far in this discussion, we have expanded, we've taken the list of topics, added more topics. Now we have to shrink, and we have to say like, okay, let's make it very simple. One project that, let's say we've already decided, okay, someone can raise their hand if they disagree. We've decided that defining gene panels and integrating across gene panels is one thing that we need to do as a community. Is that fair to say that's a conclusion from today's discussion? Okay. And then also algorithms in a broad space of imputing / integrating single cell and spatial data is also an essential thing. Now then we can go deeper into which particular algorithm and which particular algorithmic question, but those two things, let's say, is essential for the field. I see The people who suggested it are nodding. I'm assuming everyone else agrees as well, unless. Okay, then if we go beyond those two, and you look at this list at the bottom, which one thing do you think should be a project or a jamboree, or, or a hackathon or something like that, that we target and then we say, okay, the Spatial Forum has come together as a community and designed this SOP or this algorithm for this challenge that our community faces. Any bids? Anyone wants to raise their hand and make a case for one of these things? I'll give you a couple of minutes. Sheila.
I want to upvote Mo's point about defining what is a niche, what is a spatial domain, and so on. So, we saw in single cell that defining an ontology and what do we mean is really helpful. So maybe we can just sort of list out all of the words that we say are related to spatial cluster, spatial this, and just list out what do we mean. And so then we can have like a nomenclature. I think that's what you were getting at.
Quantitative definition. Yeah, so I guess how do you define the niche? Because I guess when you talk to biologists, stem cell biologists, it's a very different view of what is a niche compared to.
I'm remembering the discussions 6 years ago when we said, how do you define a cell type? That discussion lasted 1 hour, and I don't think we have an answer even today, 6 years later. So
but I guess this is more like pre-processing, right? So to see like how you define the neighborhood and then how you define the case and robustness analysis, Oli mentioned, I guess.
Yeah, so I guess one is the algorithmic question of which algorithm to use to define a niche. But the other one, maybe I was interpreting it as a philosophical question, like what is a niche?
Yeah, I guess it is philosophical, but then there's also just like the methodological outcome from that, right? If I happen to use cells around me to help me define cell type, is it actually a domain? No, it's still a prediction of cell type. So I kind of want us to get like a list of words and then what do we mean when we say those words with like formulae? That would be nice.
Actually, thanks for bringing that up. There is some confusion in the field between cell type and domain. So there are algorithms that will use the neighborhood to inform me of what type of cell I am. That's still a cell type definition algorithm, I would say. But then there are algorithms that allows the niche to have many different cell types. But the niche is actually a spatial unit rather than a specific individual cell. I would say that is the useful definition of a niche. Niche has to allow, in general, a niche can have cells only of one type, but that should not be the requirement of a niche. Right? Okay, so let's say defining niches can, let's say, is a priority for sure. It's a classic spatial problem.
I really agree with the importance of finding niches. And somebody called it niche, somebody mentioned FOV or region of interest, all those words meaning some specific areas of the sample that would correlate with probably clinical outcome, hopefully. So some automatic method that could automatically identify which region would have some clinical meaning. That is the most important thing to me, and it is not solve the problem at all. And if you consider, so far most analysis is based on whole slide image. So the getting output from whole slide image would attenuate the real signal. So you have to find which specific areas to focus. So we need there is no consensus how to identify, how to define. So we need some effort, serious efforts to them.
That's great. What I'm thinking about as I was hearing your question was extracting salient features. Like when a pathologist looks at a tissue, they'll say, okay, that's where the blood cell is damaged. Okay, I see that the macula is damaged, right? And so they'll zoom in on the macula there. I guess that's what you're referring to, looking at the entire image to extract the salient features, say, in a disease sample. And perhaps we can define that as a form of, yeah, maybe not. I was going to say niche detection, but actually it's okay, I would say spatial dysregulation. Maybe it comes under the spatial dysregulation question, because that's what the pathologist is looking for, a spatial region where something is wrong compared to the healthy cell. So I think that is a very important question. And I'm glad you brought that up, because so far all the challenges we've been discussing, which is gene panels, single cell spatial concordance, niche defining, are very general. They fit under this, right? But we do need to achieve benefit. We do need to move downstream of that. As I mentioned, disease and diversity are key goals that the Spatial Forum and HCA can help us get to. So we want to define MVPs, minimal viable products, that have something to do with disease. So I would say identifying aberrations, spatial aberrations, relative to healthy is a very nice feature we could have. Malta?
So, sorry, I was next.
Oh, sorry.
So in addition to defining niches, we need to also have a vocabulary of the features and characteristics that we use to define the niche. And to your point about diseases, we talk about disordered tissues. What does that mean, disordered? Can we be more specific about how we're describing these topological features that we're using to describe disease or describe niches or whatever? So I'm an ontologist, and so vocabulary is near and dear to my heart. And so having vocabularies to describe these properties, I think, can be quite valuable for the community.
Certainly, and as you're saying that, I was remembering the CAP, the Cell Annotation / Community Annotation Platform, and it's exactly the thing. You want to have some explanation for why you call this cell type this cell type. So now you want to have some explanation for why you said that tissue is dysregulated, some explanation for some interpretable definition of a niche beyond, I will give you 500,000 weights in a neural network, and that's my definition of a niche, right? Yeah.
So, the way I understood the point before about kind of features that are associated with outcome is actually very close to like what niche detection, I think what Sheila mentioned as well. This kind of, I think it's also there on biomarker discovery associated with diagnoses and outcomes. This biomarker could be just a repeatable pattern, which is of different cell types, which would be a niche. And so if we can kind of pinpoint that, I think we'd cover the disease aspect really well with what I think Sheila and Mo were saying, really kind of defining how we, and I also don't think it'll be one definition. I think niches are going to be at different scales, different uses, and so.
Just like cell types don't have one
exactly.
definition.
So I completely appreciate your point before about the cell type discussion.
Yeah. But yeah, even higher level of
But I think also the ability to deploy these different definitions to different data sets, will be really very useful. That when standardizes this, maybe industrializes even this sort of niche deployment would be quite useful. As a suggestion for the MVP, I think picking up on some comment that Dana made earlier, which I think was well placed, it could be useful that we have sort of one, very high level, close to the clinical outcome. I think there's a lot of agreement here in the room, as far as I can say, that niches or spatial aberrations, we can work a bit in the vocabulary, is sort of the broader area. In the other area might be the low-level processing, to avoid a sort of garbage in, garbage out. And if one has these two pillars, they can cross-fertilize and they can go in parallel. And then at some point, one can even look at how the niche definition is a function of the low-level processing. And there will be some components that certainly there will be interplay. Just as a suggestion, maybe others can pick up.
Very, very quick comment from Luis. Perhaps a NAP, niche annotation platform, would be useful.
Awesome. Yes, that's logical inference from that. Yeah, that would be great. If niches are things that are reproducible, that interpretable, and that people can talk about beyond that one paper that we published, there has to be a niche annotation project. I think that's fair to say. It sounded fantastic, but I think it's very sensible.
Hi, Shyam. I think in this as well, it's worth speaking to our pathology colleagues, because some niches that we would define from pathology, like a granuloma, for example, which have clear clinical importance for diagnostics. In inflammatory bowel disease, if it's granulomatous, it's Crohn's, it's not ulcerative colitis. So that's a clear structure. It's not disorganized tissue, it's a clear tissue structure. So I think discussion, interaction with pathologists for what they think is meaningful would be important in niche definition.
Thanks so much for that, Gary. So that just comes up in all our spatial omics projects when we work with clinicians. So I'm just going to put in there on your behalf and my behalf that integration with pathology data and pathology annotations as a problem statement, as some course problem statement that's we do need to address. Otherwise, we're just ignoring the entire history of how people have spatially characterized tissues. We cannot do that. I'm going to put a plug in for cell segmentation, which a lot of people have talked about, and let's just stipulate that that's also a key priority. And that covers a lot of what we have here and goes beyond what we have here, which is amazing. That was the purpose of this discussion. We still have 5 minutes. So would anyone like to nominate another one of these MVPs that has not been? Talk starts in 5 minutes, then we should close. Yeah. Okay, anyone in 10 seconds, someone want to make a point quickly before we close? Yeah.
I'm not really sure cell segmentation is that important because we are having experimental techniques that could determine the membrane boundaries experimentally. And the other thing is, even we have been doing good success with Visium data that does not have any cell boundary at all. But we use deconvolution.
Right. That's a whole different strategy. Yes.
Quite doubtful. So I would say cell segmentation may not be that our top priority, even though it can be important. But we have experimental methods to determine the boundaries at this point.
In which case, we should benchmark these new cell segmentation methods to see if they're really good enough.
Yeah.
Okay, and you have the last word. Thank you for that. Yeah. Thank you so much, everyone. And please email us if there's anything left that's undiscussed. Thanks again.